Photosynthesis in higher plants, Part 2. What is light reaction? Light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH are included in light reactions or the photochemical phase. In the process, several complexes are involved. Within the photosystem 1 and photosystem 2, the pigments are organized into two discrete photochemical light harvesting complexes. These are not named in the sequence in which they function during the light reaction, but these are named in the sequence of their discovery. Hundreds of pigment molecules bound to proteins constitute the LHC. All the pigments except one molecule of chlorophyll A forming a light harvesting system called antennae are present in each photosystem. By absorbing different wavelengths of light, these pigments help to make photosynthesis more efficient. The reaction center is formed by a single molecule of chlorophyll A. In both the photosystems, the reaction center is different. The reaction center chlorophyll A in photosystem 1 has an absorption peak at 700 nm and hence is called P700, while in photosystem 2 it is called P680 as the absorption maxima is at 680 nm. The electron transport. The reaction center chlorophyll A in photosystem 2 absorbs 680 nm wavelength of red light, due to which electrons become excited and jump into an orbit farther from the atomic nucleus. An electron acceptor picks up these electrons and passes them to an electrons transport system consisting of cytochromes. In terms of an oxidation reduction or redox potential scale, this movement of electrons is downhill. As they pass through the electron transport chain, the electrons are not used up but are passed on to the pigments of photosystem PS1. Simultaneously, in the reaction center, the PS1, when the electrons receive red light of wavelength 700 nm, they become excited and are transferred to another acceptor molecule which has a greater redox potential. Again, the electrons are moved downhill to a molecule of energy-rich NADP ion. NADP ion is reduced to NADPH plus hydrogen ion by the addition of these electrons. This whole scheme of transfer of electrons starting from the PS2 to the acceptor uphill to PS1 down the electron transport chain, excitation of electrons transfer to another acceptor and finally to NADP ion, causing it to be reduced to NADPH plus hydrogen ion downhill, is called the Z scheme because of its characteristic shape. When all the carriers are placed in a sequence, this shape is formed on a redox potential scale, splitting of water. You would then ask, how are the electrons continuously supplied by PS2? From photosystem 2, the electrons that were moved must be replaced. This is achieved by the splitting of water, due to which electrons are available. Associated with the PS2 is the splitting of water. Water is split into H plus ion, O and electrons. This creates oxygen, which is one of the net products of photosynthesis. Photosystem 2 provides the electrons needed to replace those removed from photosystem 1. We need to emphasize here that with the photosystem 2, the water splitting complex is associated, which on the inner side of the membrane of the thylakoid is physically located. Then, where is the oxygen and protons formed likely to be released in the lumen or on the outer side of the membrane? Cyclic and non-cyclic photophosphorylation. From oxidizable substances, living organisms have the capability of extracting energy, and in the form of bond energy, this is stored. In their chemical bonds, special substances like ATP carry this energy. The process by which the cells synthesize ATP is named phosphorylation. Photophosphorylation in the presence of light is the synthesis of ATP from ADP and inorganic phosphate. A process called non-cyclic photophosphorylation occurs when the two photosystems work in a series, first photosystem 2 and then photosystem 1. As seen earlier in the Z scheme, the two photosystems are connected through an electron transport chain. By this kind of electron flow, both ATP and NADPH plus H plus ion are synthesized. The electron is circulated when only photosystem 1 is functional within the photosystem, and due to cyclic flow of electrons, the phosphorylation occurs. The stroma lamellae is a possible location where this could be happening. The stroma lamellae membranes lack PS2 as well as NADP reductase enzyme, while the membrane or lamellae of the grana have both PS1 and PS2. The excited electron is cycled back to the PS1 complex through the electron transport chain and does not pass on to NADP ion. Hence, the cyclic flow results only in the synthesis of ATP but not of NADPH plus H plus ion. When only light of wavelengths beyond 680 nm are available for excitation, cyclic photophosphorylation occurs. Chemiosmotic hypothesis. How exactly is ATP synthesized in the chloroplast? Let us now try and understand. To explain the mechanism, the chemiosmotic hypothesis has been put forward. As in respiration, in photosynthesis also linked to the development of a proton gradient across a membrane is ATP synthesis. This time there is thylakoid membranes. There is one difference though towards the inside of the membrane, that is in the lumen is the accumulation of protons. In respiration, when electrons move through the ETS, electrons accumulate in the intermembrane space of the mitochondria. What causes the proton gradient across the membrane? Let us understand. The processes that take place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop need to be considered again. A. Since on the inner side of the membrane, splitting of the water molecule takes place, and by the splitting of water, the protons or hydrogen ions that are produced accumulate within the lumen of the thylakoids. B. As the protons are transported across the membrane, electrons move through the photosystems. This happens because towards the outer side of the membrane, the primary electron acceptor is located and transfers its electron to the H carrier and not to an electron carrier. Hence, while transporting an electron, this molecule moves a proton from the stroma. On the inner side of the membrane, when this molecule passes on its electron to the electron carrier, the proton is released into the inner side or the lumen side of the membrane. C. On the stroma side of the membrane, the NADP reductase enzyme is located. Protons are necessary for the reduction of NADP ion to NADPH plus H plus ion along the electrons that come from the acceptor of electrons of photosystem 1. These protons from the stroma are also removed. Hence, protons in the stroma within the chloroplast decrease in number while there is accumulation of protons in the lumen. This creates a measurable decrease in pH in the lumen as well as a proton gradient across the thylakoid membrane. Why are we so interested in the proton gradient? This gradient is important because the breakdown of this gradient leads to the release of energy. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the F0 of the ATPase. In the ATPase enzyme, there are two parts. One is the F0 which is embedded in the membrane, and a transmembrane channel is formed that carries out facilitated diffusion of protons across the membrane. The other portion protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma and is called F1. To cause a conformational change in the F1 particle of the ATPase, the breakdown of the gradient provides enough energy which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATPase. Within the thylakoid lumen, to create a gradient or a high concentration of protons, energy is used to pump protons across the membrane. Back across the membrane, ATPase has a channel that allows diffusion of protons. To activate ATPase enzyme, this releases enough energy that catalyzes the formation of ATP. Produced by the movement of electrons along with the NADPH, immediately the ATP will be used in the biosynthetic reaction taking place in the stroma, responsible for synthesis of sugars and fixing carbon dioxide. Where are the ATP and NADPH used? We learned that ATP, NADPH, and oxygen are the products of light reaction. Of these, ATP and NADPH are used to drive the processes leading to the synthesis of food, more accurately sugars, while oxygen diffuses out of the chloroplast. This is the biosynthetic phase of photosynthesis. On the presence of light, this process does not depend directly, but it depends on the products of the light reaction, that is NADPH and ATP besides carbon dioxide and water. How this could be verified, you may wonder, but it is simple, and biosynthetic process continues for some time immediately after light becomes unavailable and then stops. The synthesis starts again if then the light is made available. Hence, we can say that calling the biosynthetic phase as a dark reaction is a misnomer. Discuss this amongst yourself. How are the ATP and NADPH used in the biosynthetic phase? Let us see now. We saw earlier that to produce CH2ON or sugars, the carbon dioxide is combined with water. It was of interest to the scientists to find out when carbon dioxide is taken into a reaction or fixed, what was the first product formed, or rather how is reaction proceeded. Among the several efforts to put radioisotopes to beneficial use just after World War II, the work of Melvin Kelvin is exemplary. In algal photosynthesis studies done by him, the use of radioactive C14 led to the discovery that a 3-carbon organic acid was the first carbon dioxide fixation product. He also contributed to working out the complete biosynthetic pathway. It was hence called Kelvin cycle after him. 3-phosphoglyceric acid, or in short PGA, was the first product identified. How many carbon atoms does it have? Scientists also tried to know whether the first product of carbon dioxide fixation in all plants is PGA, or in other plants whether any other product was formed. Over a wide range of plants, experiments conducted led to the discovery of another group of plants where the first stable product of carbon dioxide fixation again was an organic acid, but it had 4 carbon atoms in it. This acid formed was identified to be oxaloacetic acid or OAA. Carbon dioxide assimilation during photosynthesis since then was said to be of two main types: those plants in which C3 acid was the first product of carbon dioxide fixation, that is, the C3 pathway, and those in which C4 acid was the first product, that is, the C4 pathway. Other associated characteristics were shown by these two groups of plants that we will discuss later. The primary acceptor of carbon dioxide. A molecule would have how many carbon atoms which after accepting carbon dioxide would have 3 carbons? Very unexpectedly, the study showed that a 5-carbon keto sugar was the acceptor molecule, and it was ribulose bisphosphate. Did any of you think of this possibility? Do not worry. The scientists conducted many experiments and took a long time to reach this conclusion. Since the first product was a C3 acid, the scientists also believed that the primary acceptor would be a 2-carbon compound. To identify a 2-carbon compound, they spent many years before they discovered the 5-carbon RUBP, the Kelvin cycle. The whole pathway was then worked out by Kelvin and his co-workers, and they showed that the pathway operated in a cyclic manner and the RUBP was regenerated. Let us now see where the sugar is synthesized and how the Kelvin pathway operates. Let us understand very clearly that in all photosynthetic plants, the Kelvin pathway occurs, and whether they have C3 or C4 or any other pathways, it does not matter. The Kelvin cycle, for ease of understanding, can be described under 3 stages: carboxylation, reduction, and regeneration. 1. Carboxylation. The fixation of carbon dioxide into a stable organic intermediate is carboxylation. In the Kelvin cycle, carboxylation is the most crucial step where, for the carboxylation of RUBP, carbon dioxide is utilized. The enzyme RUBP carboxylase catalyzes this reaction, which results in the formation of 2 molecules of 3-PGA. Since oxygenation activity is also present in the enzyme, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. 2. Reduction. This consists of a series of reactions which led to the formation of glucose. Per carbon dioxide molecule fixed, the steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction. For the removal of 1 molecule of glucose from the pathway, the fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required. 3. Regeneration. If the cycle is to continue uninterrupted, regeneration of the carbon dioxide acceptor molecule RUBP is crucial. To form RUBP, the regeneration steps require 1 ATP for phosphorylation. Hence, 3 molecules of ATP and 2 of NADPH are required for every carbon dioxide molecule entering the Kelvin cycle. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. 6 turns of the cycle are required to make 1 molecule of glucose. To make 1 molecule of glucose through the Kelvin pathway, work out how many ATP and NADPH molecules are required. C4 pathway. Plants that have the C4 pathway are adapted to dry tropical regions mentioned earlier. Though these plants' first carbon dioxide fixation product is C4-oxaloacetic acid, they use the C3 pathway or the Kelvin cycle as the main biosynthetic pathway. In what way then are they different from C3 plants? C4 plants are special. These plants have special leaf anatomy. They can tolerate higher temperatures to high light intensities. They show a response. A process called photorespiration is absent and productivity of biomass is greater. Let us understand these one by one. Study vertical sections of the leaves, one of a C3 plant and the other of a C4 plant. Do you notice the differences? Do both have the same types of mesophils, or around the vascular bundle sheath do they have similar cells? In the C4 pathway, plants, the large cells around the vascular bundles are called bundle sheath cells, and the leaves with such anatomy are said to have Kranz anatomy. Kranz means wreath and is a reflection of the arrangement of cells. Around the vascular bundles, the bundle sheath cells may form several layers, and they are characterized by having a large number of chloroplasts to gaseous exchange. The thick walls are impervious and no intercellular spaces. To observe the Kranz anatomy and the distribution of mesophil cells, you may like to cut a section of the leaves of C4 plants, maize or sorghum. For you to collect leaves of diverse plants species around you, it would be interesting, and then cut vertical sections of the leaves. Observe under the microscope around the vascular bundles. Look for the bundle sheath. In the C4 plants, the presence of the bundle sheath would help you identify. In the figure, now study the pathway. This pathway is again a cyclic process and has been named the Hatch and Slack pathway. By listing the steps, let us study the pathway. A 3-carbon molecule, phosphoenol pyruvate or PEP, is the primary carbon dioxide acceptor and is present in the mesophil cells. PEP carboxylase or PEPase is the enzyme responsible for this fixation. It is important to register that Rubisco enzyme is absent in the mesophil cells. In the mesophil cells, the C4 acid OAA is formed. Other 4-carbon compounds like malic acid or aspartic acid are then formed in the mesophil cells itself, which are then transported to the bundle sheath cells. In the bundle sheath cells, carbon dioxide and the 3-carbon molecule are released when these C4 acids are broken down. To the mesophil, the 3-carbon molecule is transported back, where again it is converted to PEP, thus completing the cycle. In the bundle sheath cells, the carbon dioxide released enters the C3 or the Kelvin pathway, a pathway common to all plants. The bundle sheath cells lack PEPase but are rich in an enzyme ribulose bisphosphate carboxylase oxygenase or Rubisco. Thus, the Kelvin pathway, the basic pathway that results in the formation of the sugars, is common to C3 and C4 plants. Did you note that in all the mesophil cells of the C3 plants, the Kelvin pathway occurs? In the C4 plants, the Kelvin pathway takes place only in the bundle sheath cells but does not take place in the mesophil cells. Photorespiration. Let us try and understand a process that creates an important difference between C3 and C4 plants, photorespiration. We have to know a little bit more about the first step of the Kelvin pathway, the first carbon dioxide fixation step, to understand photorespiration. This is the reaction where to form two molecules of 3-PGA, RuBP combines with carbon dioxide, that is catalyzed by Rubisco. The most abundant enzyme in the world is Rubisco, where its active site can bind to both carbon dioxide and oxygen, hence the name. Can you think how this could be possible? Rubisco for carbon dioxide has a much greater affinity than for oxygen. If this were not so, imagine what would happen. This binding is competitive. The relative concentration of oxygen and carbon dioxide determines which of the two will bind to the enzyme. Carbon dioxide fixation is decreased in C3 plants as some oxygen does bind to Rubisco. Instead of being converted to two molecules of PGA, RUBP binds with oxygen to form one molecule and phosphoglycolate in a pathway called photorespiration. There is neither synthesis of sugars nor ATP in the photorespiratory pathway. With the utilization of ATP, it rather results in the release of carbon dioxide. There is no synthesis of ATP or NADPH in the photorespiratory pathway. Therefore, photorespiration is a wasteful process. Photorespiration does not occur in C4 plants. This is because at the enzyme site they have a mechanism that increases the concentration of carbon dioxide. This takes place in the bundle cells when the C4 acid from the mesophyll is broken down to release carbon dioxide, which results in increasing the intracellular concentration of carbon dioxide. This in turn ensures that the Rubisco functions as a carboxylase and minimizes the oxygenase activity. C4 plants lack photorespiration, due to which the productivity and yield of these plants are better. In addition to higher temperatures, these plants show tolerance, factors affecting photosynthesis. It is necessary to understand the factors that affect photosynthesis. In determining the yield of plants, including crop plants, the rate of photosynthesis is very important. Photosynthesis is under the influence of both internal and external factors. The plant factors include the age, size, number, orientation of leaves, mesophyll cells, chloroplasts, the amount of chlorophyll, and internal carbon dioxide concentration. In the plant, the internal factors are dependent on the growth of the plant and the genetic predisposition. The availability of sunlight, temperature, carbon dioxide concentration, and water are the external factors that affect photosynthesis. Simultaneously, as a plant photosynthesizes, all these factors will affect the rate of photosynthesis. Several factors, though, interact and simultaneously affect photosynthesis or carbon dioxide fixation. One factor usually is the major cause or is the one that limits the rate. Hence, by the factor available at suboptimal levels, the rate will be determined at any point. Blackman's law of limiting factors comes into effect when several factors affect any chemical process. This states the following. If more than one factor affects a chemical process, then by the factor which is nearest to its minimal value, the rate will be determined. If its quantity is changed, it is the factor which directly affects the process. For example, if the temperature is very low, the plant may not photosynthesize despite the presence of the green leaf, optimal light and carbon dioxide conditions. This leaf will start to photosynthesize if given the optimal temperature. Light. While discussing light as a factor that affects photosynthesis, we need to distinguish between light quality, light intensity, and the duration of exposure to light. At low light intensities, there is a linear relationship between incident light and carbon dioxide fixation rates. Gradually, the rate does not show further increase at higher light intensities as other factors become limiting. What is interesting to note is that at 10% of the full sunlight, light saturation occurs. Light is rarely a limiting factor in nature except for plants in shade or in dense forests. Beyond a point, increase in incident light causes the breakdown of chlorophyll and photosynthesis decreases. Carbon dioxide concentration. The major limiting factor for photosynthesis is carbon dioxide. In the atmosphere, the concentration of carbon dioxide is very low. Up to 0.05% increase in concentration can cause an increase in carbon dioxide fixation rates. Beyond this, over longer periods, the level can become damaging. To the carbon dioxide concentration, the C3 and C4 plants respond differently. Neither group responds to high carbon dioxide conditions at low light conditions. At high light intensities, increase in the rates of photosynthesis is seen in both C3 and C4 plants. What is important to note is that at about 360 μl per liter, the C4 plants show saturation, while in C3 plants saturation is seen only beyond 450 μl per liter, and they respond to increased carbon dioxide concentration. Thus, to the C3 plants, current availability of carbon dioxide levels is limiting. The fact that C3 plants respond by showing increased rates of photosynthesis to higher carbon dioxide concentration, leading to higher productivity, has been used for some crops in the greenhouse, such as bell pepper and tomatoes. They are allowed to grow in an atmosphere enriched in carbon dioxide, which leads to higher yields. Temperature. The dark reactions, being enzymatic, are controlled by temperature. Though the light reactions are affected to a much lesser extent, they are also sensitive to temperature. The C4 plants show higher rate of photosynthesis as they respond to higher temperatures, while C3 plants have a much lower temperature optimum. In different plants, the temperature optimum for photosynthesis depends on the habitat that they are adapted to. A higher temperature optimum is seen in tropical plants than the plants adapted to temperate climates. Water. In the light reaction, even though water is one of the reactants as a factor, the effect of water is more through its effect on the plant rather than on photosynthesis directly. The stomata closest to water stress, hence the availability of carbon dioxide reduces. Besides leaves wilt due to water stress, the surface area of the leaves and their metabolic activity is reduced as well. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.